Hello, my name is Siti Nurakilah binti Zali, and this video presents my final year project titled Molecular Identification of Klebsiella pneumoniae isolated from agricultural soil samples. The overview of what I will be presenting includes background of study, problem statement, objectives, methodology, results and discussion, and conclusion and recommendation. For the background of study, Klebsiella pneumoniae is a human opportunistic pathogen that inhabits non-clinical environments including soil, water, and plant-associated conditions as stated by Mahdi et al. 2024. In Malaysia, pneumonia remains one of the leading causes of illness, with Klebsiella pneumoniae identified as one of the primary bacterial agents. This bacterium is present in agricultural soil due to its natural presence in the rhizosphere, where it plays a role in nutrient cycling and may serve as a potential environmental reservoir of clinical concern. The problem statement of this research is that Klebsiella pneumoniae is a harmful bacterium that can cause serious infections such as pneumonia, bloodstream infections, and also urinary tract infections in humans. Frequent human interaction with soil, such as during farming, crop handling, and the use of organic fertilizers, can increase the risk of spreading this bacterium. Although it has been widely studied in clinical settings, there is still limited research on its presence, role, and also characteristics in agricultural soil. So the objective of this research are, first, to characterize the presence of Klebsiella pneumoniae in agricultural soil samples through morphological observation and biochemical tests. And the second one is to identify the presence of Klebsiella pneumoniae in the soil sample through polymerase chain reaction or PCR. The methods used in this research is the first one. Soil samples were collected from an agricultural farm and placed in sterile containers. Then a 10-gram portion of the soil was suspended in 9 ml of sterile saline solution and vortexed vigorously for 2 minutes, followed by a several dilution. The liquids were cultured on McConkey agar and incubated at 37 degrees Celsius for 18 to 24 hours. Then, gram staining was performed to observe the shape and the size of bacterial cells under the microscope. Next, biochemical tests such as indole tests, citrate tests, and urease tests were carried out to characterize and detect the presence of Klebsiella pneumoniae. And lastly, PCR was performed to amplify the 16S rRNA target gene, and the PCR product was sent for sequencing to confirm the identity of Klebsiella pneumoniae. After incubation of McConkey agar, colonies with typical Klebsiella pneumoniae morphology were observed. They appeared large, round, smooth, pink and mucoid, indicating lactose fermentation and capsule production. So the pink colors results from acid production, while the mucoid texture is due to the bacterium stick capsule. No gram-positive was observed, confirming McConkey agar selectivity. Out of 14 initial isolates, only 4 consistently showed lactose fermenters, which could indicate the characteristic of targeted bacteria. Gram staining was performed on the isolated colonies to observe the gram reaction. Under the microscope, all 4 isolated cells appeared pink and rod-shaped, confirming they were gram-negative bacilli, a characteristic of Klebsiella pneumoniae. These rods were seen either singly or in short chains, a common feature of Enterobacteriaceae family. For comparison, the reference strain ATCC Klebsiella pneumoniae was also tested, and its morphology was found to be consistent with the environmental isolates. Next, biochemical test performed was indole test, which checks if a bacterium can break down tryptophan into indole using the enzyme tryptophanase. After adding convex reagent, a red or pink ring indicates a positive result. In this study, isolates 1, 2, and 3 showed a negative indole test, meaning there was no color change, while isolate 4 showed a positive reaction. This suggests that isolates 1, 2, and 3 are likely Klebsiella pneumoniae, as it typically cannot produce indole. The indole test also helps to differentiate Klebsiella pneumoniae from related bacteria like Klebsiella oxytoca and also E. coli, which are usually indole positive. Next, the citrate test checks if a bacterium can use citrate as its only carbon source using an enzyme called citrate permease. A positive result is shown when the cymo-citrate agar changed from green to blue due to the alkaline byproducts. In this study, isolates 1, 2, and 3 showed a positive results, while isolates 4 was citrate negative with no color change. This result supports that isolates 1, 2, and 3 may be Klebsiella pneumoniae, which is usually citrate positive. The citrate test also helps to differentiate it from other citrate-negative soil bacteria like Pentia and also Cosaconia species. Next, the urease test checks if a bacterium can break down urea into ammonia and carbon dioxide, which raises the pH. A positive result is seen when the urea agar changed from orange to bright pink, showing alkalinity. In this study, isolates 1, 2, and 3 showed a positive results, while isolate 4 was urease negative with no color change. These results support that isolates 1, 2, and 3 again may be Klebsiella pneumoniae, which is usually urease positive. The urease test is also helpful for distinguishing Klebsiella pneumoniae from other Enterobacter species, which are often urease negative or weak producers. PCR was done on isolates 1 and 2 using 16S rRNA primers to confirm their molecular identity. The PCR products were analyzed using agarose gel electrophoresis, and clear bands were seen in the positive lanes at the expected size, confirming successful amplification on the target gene. Negative results, the negative control, showed no band, proving there was no contamination and the reaction was specific. These results validate the overall isolation and detection process. After the successful PCR, the DNA from isolate 2 was sent to Sanger sequencing to confirm its identity. The sequence was compared using BLAST from the NCBI database. The results showed 97.56% similarity and 85% query coverage with a known Klebsiella pneumoniae strains EB6THQ. This high similarity confirms that the isolate is genetically related to Klebsiella pneumoniae, supporting its identification from agricultural soil. In conclusion, Klebsiella pneumoniae was successfully identified from agricultural soil using biochemical tests and PCR analysis. These findings show that environmental sources like agricultural soil can harbor harmful bacteria with clinical importance. For future studies, it is recommended to test for antibiotic-resistant AMR genes and virulence genes to better understand the potential risks this isolated may pose. That's all. Thank you for listening.